It's easy to think of the cell membrane as this boring structure that's just sitting there. Well, that's totally not the case. Pieces of the membrane are in constant motion, and there are a lot of different types of pieces. This video is 6.5 on the fluid Mosaic model of the cell membrane. We're moving on to these 3 goals in this video to outline some membrane basics. Pause the video to check them out. The concept of the membrane as a fluid mosaic is pretty straightforward if you just look at what the words are telling you. Fluid refers to the fact that the subunits of the membrane are in constant motion. They slide right past one another. And mosaic is just like in art. Remember one of these, where there's a picture made by putting smaller pieces or tiles together? A membrane is a mosaic with many different types of smaller pieces. Look at all of these different types of pieces in this model drawing of a membrane. The mosaic components of membranes are mostly lipids and proteins. Of course, phospholipids are the main membrane component, but you can see cholesterol on this list, and cholesterol is interspersed in between the phospholipids as well. And finally, there are some glycolipids, and you can see one right here, which are lipids that have 2 fatty acids and some sugar groups attached instead of a 3rd fatty acid or a phosphate. There are also many different protein mosaic pieces that make up the membrane as well. Glycoproteins are proteins with sugars attached, like this one right here. Some glycoproteins are also adhesion proteins, which allow cells to stick to other cells or the cell surroundings. See how this sugar is allowing this cell to stick to these environmental pieces? Some other membrane proteins include receptor proteins that bind to signaling molecules and channel proteins like this one right here, which create tunnels to allow substances to pass through the membrane. Well, let's talk a little more about those lipid components. To review, phospholipids are modified triglycerides. You can see this glycerol backbone with 2 fatty acids attached by ester bonds and 1 phosphate group in the 3rd slot of glycerol. Phospholipids are typically drawn with a circle to represent the phosphate group and 2 lines or 2 tails to represent the fatty acids. Biochemically, the phosphate head group is hydrophilic because of its negative charge, and the nonpolar nature of the fatty acid tails make the tails of the phospholipid hydrophobic. When one side of a molecule is hydrophilic and the other side is hydrophobic, and this is called amphipathic. Phospholipids are amphipathic. As a result, phospholipids do something really cool. When phospholipids are placed in a watery environment, like what this blue is supposed to represent, the phospholipids spontaneously form a bilayer. This is because NLS, nature loves stability. Let me explain. The hydrophobic or nonpolar fatty acids hate being by water, and so the tails are more stable being by each other, like dissolves like. Similarly, the charged hydrophilic phosphate groups like being towards a watery environment. In cells, there are watery environments inside of the cell, the cytoplasm, and outside of the cell too. And that's why a lipid bilayer is so stable. As a bilayer, the hydrophilic parts get to be around water, and the hydrophobic parts get to be around each other, away from water. Besides lipids, the other mosaic pieces are proteins. Membrane proteins can have so many different cool functions. Transport proteins help molecules pass through the membrane. There are membrane-bound enzyme proteins that help speed up chemical reactions. This protein's a receptor that's binding to a yellow signaling molecule, a signal from another cell. These proteins are allowing 2 cells to stick together, and this is a glycoprotein. It's like an ID card where the shape of the sugar tells the cell's identity. And finally, in this diagram, there's an adhesion protein that helps the cell attach itself to the extracellular matrix outside of the cell. Membrane proteins can also be peripheral membrane proteins or integral membrane proteins. Integral membrane proteins interact with the fatty acids. You can say that integral membrane proteins are integrated into the membrane, like this one interacting with the fatty acids here, and this one going all the way through. And so many integral membrane proteins are also transmembrane proteins, like these 2 guys right here, meaning that they go all the way through from the cytoplasmic side all the way through to the extracellular side. In contrast, peripheral membrane proteins are only loosely associated with the membrane. often only interacting with the phosphate heads. Next, I want to talk about what it means for the membrane to be fluid. Fluid means that the membrane components move, but membrane components can only move in a certain way. Now check out these phospholipids right here. You could see the lateral movement is quite frequent according to this diagram, sliding back and forth. In other words, the phospholipids can drift and slide past one another. It's very rare, though, that phospholipids would move by flip-flopping and changing directions. So a phospholipid that's pointing to the cytoplasm will pretty much always stay pointed in that direction. Now, the reason why flip-flopping is so rare is because of the biochemistry of phospholipids. In order to flip-flop, the negatively charged phosphates would have to pass through the hydrophobic fatty acid core of the bilayer, and that would just be very unfavorable. Now, there are some factors that help keep the membrane fluid and allow that lateral movement. The first is using unsaturated fats in the tails of phospholipids. Remember, an unsaturated fat has a carbon-carbon double bond in the carbon backbone of the fatty acid that introduces a kink or a bend in the fatty acid, and having those bends makes it harder for the fatty acids to pack together into a solid, and that makes the membrane more fluid, allowing membrane components to slide past one another. Having cholesterol mixed into the plasma membrane in yellow here, and here, in 3 different models here, cholesterol also helps with membrane fluidity. And you can see the effect that cholesterol has on preventing the phospholipids from packing together too closely. Another functional advantage of cholesterol is that cholesterol helps the membrane be a better barrier by acting as an additional group of hydrophobic molecules to repel polar and charged particles from getting through. So let's talk about that barrier function a little bit more. That's the main function of the membrane, to keep the inside of a cell separated from the outside environment. But here's the deal. A cell still needs to move substances in and out. And so a cell must be selectively permeable. Selective permeability means that only some substances can pass through the membrane. The biochemical properties of the cell membrane give the cell some control of what passes through. The double layer of hydrophobic fatty acids makes a membrane really impermeable to charged polar molecules. And that's just because hydrophobic fatty acids and hydrophilic things really don't mix. And you can see from this chart right here that nonpolar molecules can actually pass through the membrane pretty easily. This is because nonpolar molecules don't have a problem getting through the nonpolar fatty acids, like dissolves like. But molecules that are polar have a really hard time. Still, though, small molecules that are polar can make it through a little easier than larger polar molecules or ions, just because of how membrane pieces are packed closer together. But polar or charged substances still need to get into the cell. Think about all these things listed here: water, amino acids, glucose, ammonium, and ions. A cell couldn't live without moving those substances in and out. So to solve this problem, the membrane has the mosaic pieces of protein channels. Now, a cool thing about protein channels is that each channel is specific to the type of molecule the channel will let go through. So, for example, there are water channels called aquaporins, and there are specific glucose channels too. The reason these channels are proteins is because the biochemical diversity of proteins. Remember all of these different R groups with all these different biochemical properties? We've got polar, electrically charged, nonpolar. Well, these biochemical properties are essential for a channel protein to function properly. Let me explain. Here's a typical channel protein, and think about the complex biochemical interactions the channel protein has to manage. The channel protein has to interact with the watery extracellular environment out here, and then a channel protein has to pass through the hydrophobic fatty acid tails and finally interact with the watery cytoplasm inside of the cell. And on top of that, the channel protein has to create a tunnel that has compatible biochemistry with the substance that's passing through. Now, to do this, channel proteins are folded strategically so that specific R groups are positioned in specific places. Like any place where the protein is embedded in the fatty acids, like these red spots right here, there are going to be nonpolar R groups sticking out because those nonpolar R groups will play nice with these nonpolar fatty acids. Now, any place where the channel protein is facing inside or facing outside, like all these purple spots here, there are going to be polar or charged R groups sticking out. And this makes proteins perfect for the job of channel proteins, because proteins can be stable even though if there's changing biochemical patterns that the protein has to be stable with. We covered a lot of ground about membranes. First, the membrane is a fluid mosaic. There are a lot of protein and lipid pieces that make it a mosaic, and those pieces are fluid because they move laterally around each other. A membrane can be fluid because unsaturated fats in the phospholipids, as well as cholesterol, keep phospholipids from packing too close together and allow them to slide past each other easier. A membrane is a mosaic because there are a lot of different types of lipid and protein pieces that make up a membrane. The phospholipids and cholesterol make the membrane a really good barrier to polar, charged, and large molecules, but membrane proteins allow specific charged substances to pass through. This property is called selective permeability.